Okay, I wanted to use the next series of videos to talk about how membrane potential, resting potential, action potential, synapses, and the autonomic nervous system are all interrelated. It's actually a pretty beautiful thing, but it's really easy to get lost into the nitty-gritty details without seeing the larger picture. And I think really if you understand the larger picture, then it'll help you memorize the smaller details, which are, of course, what you're going to be tested on. So feel free to pause the video or to take screenshots. I know for me it really helped to kind of use a lot of resources to try and understand a concept, and once I had an idea, just draw my own diagrams and to draw my own flowcharts, which might work for you. So as we were talking about in previous videos, the way in which we can generate electrical impulses is through gradients. Okay, we need to generate these electrical impulses so that we can create some kind of action potential. The action potential will trigger the inflow or outflow of different ions. Different ions will cause another type of action potential to propagate, and ultimately we can get our target organ, whether it be skeletal muscle, whether it be smooth muscle, whether it be our sweat glands, to do what we need it to do in whatever situation. So we're going to start with the electrical impulse, and it's generating a gradient. And just to refresh your memory, we were talking about the natural order of flow inside and outside the cell. We were saying that sodium tends to move inside the cell because there's so much positive charge out here, so much sodium out here, and potassium moves out of the cell because there's more potassium inside relative to outside. This is all based on the principle that if we have our membrane, okay, this is the border of our cell, right? This is our membrane, and say, for example, that its periphery is negatively charged, and we happen to have a channel here that can pass ions through, we're more likely to pass a positively charged ion through, in which case this would be called a sodium channel. All right. And this is the concept. Now we're bringing in positive charges into this side up until a point where we saturate the number of positively charged ions that we would need, and in which case we need to stop that and then reverse the flow. Okay, so we can categorize the movement of whether it be ions or whether it be neurotransmitters into two different worlds. Okay, and the way I would kind of understand it is, on the one hand, we have voltage-gated movement, and on the other hand, we have ligand-gated. Okay. Now, voltage-gated will say that in this case we're talking about moving ions. Now, what this means is that voltage, I'm sure that professors mentioned this to you before, I believe Ohm's law, where we talk about how voltage is current times resistance. And the current that we're talking about is a current that we're generating because of this membrane potential and the difference between charges that we have here. Now, voltage gated just means that we have a gate, so we have a channel, okay, that is dependent on the voltage on one side versus the other, okay, and it also is gated, meaning it can open and it can close. So we kind of depict this through an action potential, and there's two parts of the action potential. The first part is depolarization, and if this is our action potential, this is our resting membrane potential, it's about 70 millivolts. Depolarization is this part of the action potential in which we're suddenly opening up sodium gates and we're letting sodium in. So the first part of the depolarization is the activation of sodium channels, and we're bringing sodium in. Okay, so let's say sodium is in. And then, you know, we're bringing in sodium, and we're bringing in sodium, and we're bringing in sodium, and we're, you know, making the inside of the cell pretty positively charged up until a point, let's say, okay, we're bringing the resting membrane potential up to something more positive. I think we're approaching 65 millivolts, but not quite reaching it. Anyway, we're getting to our maximum threshold, in which point we no longer need positive ions here anymore. And here we're going to close it. Which brings us to the second subpart of depolarization, which is inactivation, in which we're really not bringing anything in or out, but we're really just closing the sodium channels. And that is that part right here. The second part is, oops, I didn't make enough space, is repolarization. Sorry. Which is this part of the active potential right here, in which we are coming back down from wherever we were at, somewhere around 65 millivolts, and we're bringing it back down to actually a point that is more negative than where we actually started it. You know, you double check this, but somewhere around 80 millivolts, we hyperpolarize after depolarizing, and eventually we come back. And if you can visualize this, we're saying, okay, we already have too many positive charges in here. Let's just say that this is our potassium channel. Let's get the positive charge out, move it back out here. Oops, move it back out here so that we can balance things out again. So this is the nature of voltage-gated channels, right? Here the sodium channel is voltage-gated, here the potassium channel is voltage-gated, and it's really all based on how we are manipulating this potential. Now, the other world in which we can basically affect signals is something called the Lie-in-gated system. And it is a little bit more complicated, and it has a few more subtypes to it, which all need to be explained in detail. So here we were talking about in voltage-gated, we were creating a current through which we can try and move ions through these channels that can open and close. Now the other way is to have a ligand or something come and bind to a certain site and affect a reaction. And this is usually through either hormones, this can be through tyrosine kinase system, this can be through second messenger system, this can also be through neurotransmitters. So now let me give you the high yield points as to how they're each different. Okay, and this is just meat and potatoes of what you need to know about these signaling systems. Hormones are special. They're different from all these other systems. Because in all these other systems, what we're doing is here, tyrosine kinase, second messengers, the ultimate goal of this whole cascade is to finally phosphorylate an enzyme or phosphorylate an activating enzyme, which is going to bring about whatever effect that we want. With neurotransmitters, we're going to pass them along from synapse to synapse until they reach their target and they do what they're supposed to do. Hormones are in a whole other class of their own, in which what happens is you have a steroid hormone. Cholesterol is one, for example, if you didn't know, that is kind of floating out and about outside of the cell. It comes down, it enters the cell through, it really just goes through. You know why? Because steroid hormones are hydrophobic, or they're lipophilic. And what did we say in a previous video? We said if something's hydrophobic or if something's nonpolar, it can just go through a lipid membrane. We said oxygen will do that, carbon dioxide will do that, and fatty acids and lipids will do that. Well, these things are hydrophobic, so they're just going to pass right through the membrane. And what they're going to do is, let's say this is like a circle. This is going to bind to a steroid hormone receptor. Now, it gets fun because the steroid hormone receptor will go through the cytoplasm, right? This is the cytoplasm, and this is the nucleus. It'll go straight into the nucleus, and it'll land on DNA. And then everything will get activated. There will be transcription, there will be translation. It spits out a protein. And now this wonderful protein can do whatever action that needs to happen, whether it is to generate more of this steroid or whatnot. Okay, that is the ultimate effect. The point is that with hormones. We bring in a hormone from the outside, binds to a receptor. Together, this goes into the nucleus. It transcribes and translates its own protein, and the protein itself is active and it does something. So this is where steroid hormones are like, listen, I don't want to rely on phosphorylation reactions. I don't want to rely on synaptic junctions. I just go inside the cell and I do what I want to do, and I make more of whatever I want to make more of. That's it. It's pretty bossy. The next two ligand-based mechanisms is the tyrosine kinase protein and the second messenger system. Now, the ultimate goal for each one of these is to phosphorylate an enzyme, produce phosphorus. Why? Because phosphorus is pretty much the ultimate activating molecule. And we have these cascade reactions in order to hand off a phosphorus, which is going to activate an enzyme, and that enzyme is going to carry out whatever effect that we want. So, again, there's two mechanisms by which we can do this. In the tyrosine kinase system, and the way it's often depicted is you have this membrane. They draw it as like a T-shaped protein. Maybe it really does look like a T. I don't really know. And what happens is, I believe you have like two separate ligands that are binding to a receptor site, and the fact that they're both bound causes tyrosine amino acids that are on this protein to phosphorylate themselves. So this autophosphorylation happens, which produces phosphorus, which can be utilized by ADP or ATP, and which can be used as currency down the road. So tyrosine kinase, again, is in a class of its own because it's one of the only proteins that we talk about that has the power to generate all this phosphorus all on its own, and that's because these two things bound at this receptor site at the same time, causing this cascade of reactions. Next, things get a little tricky with the second messenger system. What is meant by a second messenger is that it's not a first messenger. So what did the first messenger do? The first messenger came down and it bound to something called G protein that is sitting on the membrane. G. Now there are several types of G proteins, okay? There is G protein Q, there's G protein I, there's G protein S, and depending on the type of G protein will determine what effect it has down the road. Now, what you should remember is that G protein I has an inhibitory effect, so everything that happens after it is meant to inhibit some function. G protein Q and G protein S tend to be not inhibitory but excitatory, so the whole point is to generate some kind of productive activity down the road. Now, these G proteins are coupled to receptors. For our purposes, it's worth knowing that some of these receptors can be an alpha receptor, can be a beta receptor, this is part of the adrenergic system, or they can be a muscarinic receptor. And we talk about muscarinic receptors usually 1 through 5. In alpha and beta, there's alpha 1, there's alpha 2. In beta, there's beta 1, there's beta 2. And we talk about muscarinic 1 through 5. So we know that the G protein can be coupled to any type of receptor. It could be an alpha 1 receptor, it could be M2 receptor, it can be a beta 2 receptor. Whatever it is, it kind of just depends on what is attached. Now, depending on what is attached depends on what cascade comes after it. So I guess it would be worth mentioning the types of G proteins. So you have GQ, you have GI, and you have GS. And just briefly, what you should know is that with the GQ receptor, this binding happens, this receptor gets triggered, and in the GQ cascade, if this was a GQ protein, what we would see is phospholipase C hydrolyzing PIP2, I believe, into IP3 and into DAG. The IP3 will cause an increase in calcium. The increase in calcium will cause an increase in calmodulin. And basically this will have a phosphorylating effect. The DAG, if I'm not mistaken, increases protein kinase C, and a protein kinase, meaning protein kind ACE, will give up a phosphorus because it's generous and the phosphorus has an activating the subsequent series of reaction. If this was